Sharing.
Good afternoon, everyone. Welcome to today's seminar presentation. It is my pleasure to introduce Kevin Yu. Kevin is originally from California, where he became intrigued by invasion ecology and conservation science. This motivated him to pursue a Bachelor's of Arts in Environmental Studies and Biology from the University of California, Santa Cruz. He is currently working on a PhD in the Applied Plant Sciences department, focusing on knotweed genetics and ecology. On a personal note, he states that he is a big old nerd at heart, evidenced by his love for all types of games, ranging from board games and video games to nerd classics such as Magic the Gathering, even going as far as working as a Magic the Gathering judge for a couple of years. Thank you, Kevin, for not letting any gaming style go left behind. And with that, I'll hand it over to you.
Thanks, James. All righty. Appreciate a very warm welcome. Hi, my name is Kevin. I'll be talking with everyone today about management of invasive knotweeds. I am a PhD student in the APS program, as James already mentioned, and my advisors are Alan Smith and Neil Anderson. So before we jump into everything about knotweed, we will go over a bit about invasive species first to kind of set the broad picture, and then go into a knotweed introduction, and then switch right into knotweed genetics, and then the objectives of my research, proposed experiments of my research, and then directions that we can kind of move forward with after this is basically done. So before we talk about knotweed, we should probably talk about invasive species first. So this is a very simple definition of invasive species. Invasive species are species that negatively affect its environment, but this is not synonymous with exotic, non-indigenous, or alien. So people have used these terms interchangeably in the past, and it's not always accurate necessarily, because native invasive species do exist. Invasive species is more synonymous with harmful species or nuisance species. And invasive species have certain traits that kind of align them all together in one group. Most of them, if not all of them, have rapid growth, rapid reproduction. They can also have high dispersal, and typically they can be phenotypically very plastic. So they can tolerate a wider range of environments compared to other species. Here's an example of kudzu in the top right corner here in Georgia. It's a vine that has invaded itself quite tremendously. Zebra mussels on the bottom right. Our Great Lakes is full of a lot of these invasives, unfortunately. And a more recent discovery is spotted lanternfly in Ohio on the bottom left here. So why should we worry about invasive species at all? Well, there's a lot of different things. So they can negatively impact ecosystems, other species, and humans, and they cause quite a bit of damage. I will list them as three general categories: ecological damage, environmental damage, and economic damage. So here's a picture of Eurasian mortar milfoil in New York. As you can see, it's kind of taken over this lake. You can't really have a boat on this lake cruising around because they would just get caught in the motors, and basically you can't really go anywhere. So we'll start first with ecological damage. So invasives can reduce native species populations, and it can also potentially drive extinctions of local species. They can also introduce pests or pathogens to the area that may not necessarily have them before, and also alter ecosystem function. But there are a lot more ecological damage that we can kind of get into, but I won't go into too much detail with this today. Here's an example of North American beavers in South America. So they're obviously not invasive in North America, but in South America, they can cause a lot of ecosystem function altering through the dams they create. Next, we have environmental damage from invasive species. So this includes either reducing habitats for other species to live in, or completely altering ecosystems themselves. So this can be caused through floods, soil erosion, or even fire regimes. So here's a picture of cheatgrass in California, which has helped fuel fire events more so than other species. Cheatgrass is non-native to California, but they are very highly flammable, obviously, as you can see here. And lastly, we have economic damage. So it costs a lot of money to remove these species. It costs a lot to clean them up, using pesticides to deter insects, other plants, what have you. And it can also result in production loss. So both in the agriculture and forestry industry can encounter losses due to invasive scenario. So with that kind of covering a little bit about invasives as a background, we will now switch over to talking about knotweed as invasive. So before we jump into the nitty-gritty of knotweed himself, we'll talk about the invasion of knotweed first. So in the history of knotweed, for the US specifically, Japanese and giant knotweed were brought over here in the 1800s as ornamentals. Both were also planted as erosion control and livestock forage, but unfortunately they have since escaped cultivation. Here's a picture of a Bohemian knotweed male in Brooklyn Center, Minnesota. As you can see, it's pretty massive. There's a minivan right next to it, but that pales in comparison to the size of this knotweed plant. And here is a distribution map, one distribution map of Japanese knotweed around the world currently. But as you can see, there is one point in Greenland, but there's no points in Australia. So this map is not necessarily the most complete either. A different map shows there's nothing in Greenland, but there's some in Australia. Basically, Japanese knotweed is a global issue at this point. It's spread pretty much to all continents, but the extent of its presence is different depending on which continent and which country you're in. The UK is especially infested with these. The US is getting to certain numbers as well, but other countries not as severe. So that brings us to management concerns of knotweed. So because knotweed can spread differently depending on if it has pollen available, it can spread either through rhizomes or through seeds, depending on what's found around it, what other knotweed species is interacting with. And removal of knotweed can be pretty involved, but also unpredictable. Usually you would want to have a combination of mechanical and chemical approach to kind of get rid of knotweed. But because some of these can be pretty massive, as you can kind of see here, it's not always guaranteed to work, and you need a systemic approach to kind of get rid of knotweed, but it's not always going to work. And knotweed can be pretty costly to manage. An estimated $2 billion per year in property control alone was calculated. And this picture of knotweed in Norway, you can see it got to the point where it can hit the top of the stop sign. They can get pretty massive. And knotweed is also pretty resilient. So Japanese knotweed can be especially hardy and can grow through cracks found in foundation or concrete. On the bottom here, you can see it's kind of growing through cracks between a brick wall. And on the right, you can see it kind of penetrating through concrete, a little gap in the concrete essentially. So knotweed itself cannot penetrate through concrete alone, but any gaps, holes it can find, it can grow there and kind of expand from there on. So we talked a lot about knotweed and how invasive it can be and how hardy it is, but what is knotweed exactly? So knotweed refers to 5 Fallopius species. You can also note, you might have heard of these as Rhinutria or Polygonum. These are all synonym genus names for knotweed. These are early successional species from Eurasia. And they have, Japanese knotweed has evolved to break through volcanic soils. You can find Japanese knotweed on volcanic slopes in Japan. Knotweed are all rhizomatous, so that means they have an underground stem, and they are also all dioecious, which means they have male and female plants, and subsequently male and female flowers. And the flowers are in the form of a panicle. which is a compounded raceme, if you know what a raceme is. But if you don't, on the top right here, you can see a picture of a panicle. Basically, there's a main stem that has no flowers growing on it, but from that stem, more branch stems kind of pop off from it, and then that's where the flowers kind of grow, essentially. And a little bit about the knotweed life cycle. So knotweed is an herbaceous perennial, so it does grow year after year. And it germinates early in spring. So in Minnesota specifically, it would be around April-ish. And it can germinate, it can flower throughout summer and fall. So it's a pretty great resource for pollinators, especially. And after the fall, it's sets seed, and then the rhizomes are dormant during winter. Here's a picture of a pretty young looking knotweed plant and the seed itself, which is an achene. See. And so let's dive a little bit into North American knotweed specifically. So 3 knotweed species are of concern for us. There's Japanese knotweed, Philopia japonica, giant knotweed, Philopia saliculensis, and Bohemian knotweed, Philopia exbohemica. Bohemian knotweed is a hybrid between giant and Japanese knotweed. As you can kind of see in the picture on the left here, giant knotweed has pretty big leaves, bigger than the size of your palm usually. Japanese knotweed, the leaves are much smaller than that, but the hybrid can vary between looking more like giant and more like Japanese. So this is a distribution map of Japanese knotweed in North America as of last month, found using ETMaps. These are all female Japanese knotweed, all female Japanese knotweed plants found in North America. They have spreads as an ornamental spread since the 1800s, but has since invaded most of the states as well as several Canadian provinces. Giant knotweed doesn't seem to be as pervasive, but there is quite a bit found still. It's not in as many Canadian provinces as Japanese knotweed, but it's still in quite a number of states. However, there doesn't seem to be as much Bohemian knotweed at all compared to giant or Japanese in the North America in general. Specifically, the South and California seems to be missing, even though both giant and Japanese occur in those areas. So here's just comparing all 3 maps together. Japanese, pretty widespread. Bohemian, not so much. So why is that? So to kind of narrow the scale down a little bit, we want to talk about knotweeds in Minnesota specifically. Why? Japanese knotweed is known as one of the world's 100, it's on the list of the 100 of the world's worst invasive alien species. Bohemian knotweed is highly plastic, meaning that it can tolerate a wide range of environments. And knotweed themselves can spread either asexually or sexually. So asexual spread is usually through rhizomes, and sexual spread usually through seeds. A knotweed plant can produce thousands upon thousands of seeds per plant. And as I said before, they can hybridize. Bohemian knotweed is a result of giant and Japanese. And Japanese, giant, and Bohemian all exist in Minnesota. So we also want to know, maybe, can they tolerate cold here? But the most important part of this slide is that it's very difficult to differentiate hybrids from Japanese or giant knotweed sometimes. So we'll jump into knotweed genetics now and talk a little bit about what has been done genetically speaking in knotweed research. But why is that important, first of all? Well, the distribution history of knotweed is kind of unclear, so it's nice to have some sort of genetic mapping to kind of tell us where it kind of came from and whether populations are the same or not, especially with Japanese knotweed, which should be all clonal, but we'll know more as we kind of ID different Japanese populations in the US. There is no known reference genome because there hasn't been that much work done on knotweed. It's been more done for the last decade or so, but still more work has to be done. And because hybridization confounds visual identification of knotweed, it's kind of important to have the DNA to kind of back that up, because you kind of need the DNA to kind of ID essentially. So just a really quick poll. I'm not timing or anything, but just take a few seconds here and see, what do you think these knotweed leaves belong to? These are all different plants. I'm not tricking you with multiple leaves of the same plant, but see if you can kind of identify which one is giant, which one is Japanese, which one is Bohemian. All right, so actually, these are all Bohemian knotweed leaves. Obviously, the leftmost one looks more like giants, the rightmost one looks more Japanese. But as you can see, there's a pretty wide range of phenotypic traits that, at least leaf morphology-wise, that Bohemian leaves can kind of take. And knotweed is also a polyploid. So a polyploid basically means that it can some species can have more than 2 sets of chromosomes. So invasive Japanese is commonly an octoploid or a tetraploid. Invasive giant knotweed is commonly a tetraploid, but Bohemian knotweed, usually being commonly a hexaploid, can also be a tetraploid. It can be an octoploid, decaploid, and aneuploid as well. So a wide range of variety for ploid level for Bohemian knotweed specifically. And in all of the genetic studies of knotweed, there have been genetic markers used in these studies. Techniques range in the past from RAPDs, random amplified polymorphic DNA, to AFLP, amplified fragment-like polymorphisms, to more recently SSR and ISSR, simple sequence repeats or inter-simple sequence repeats. But more recently, it's been more SNP stuff, so single nucleotide polymorphisms. That's been most cost-effective and cheap way to get a lot of information in. And DNA markers and chloroplast markers were primarily used. These were all basically used to determine genetic diversity between knotweed populations. And the latest SNP usage is through DART-seq. This is part of what Dallas Drazen has been doing for her master's, if you know Dallas. So DART-seq stands for Diversity Array Technology Sequencing. So this is a form of next-gen sequencing, if you know what that is, utilizing genotyping by sequencing. And it basically produces SNP data, essentially. This helps us to obtain the genetic diversity of knotweeds in Minnesota without a reference genome. And what Dallas did here was compared samples in Minnesota and Wisconsin to samples from Belgium, Poland, the UK, Japan, Western US, and Canada as well, to see if there's any differences or not. And here is a very preliminary data slide on what she found. I wouldn't use this as an end-all picture for the whole story, but this is what she had, I think, back in April, I believe. But the whole point of this is to basically visualize how populations or how species of knotweed are kind of different genetically speaking. And this is a principal coordinates analysis, essentially. This is not a PCA, this is a PCOA. The PCA, or principal coordinates analysis, visualizes dissimilarities or similarities between populations. A principal component analysis, or PCA, you just use that to reduce the dimension. So a little bit different here, but the point remains the point here is basically Japanese on the left side is a specific group compared to Bohemian in the middle, and giant on the right. That's the take home for this. And with that, let's move on to the objectives of the study. So from this research is being funded by the Minnesota Invasive Terrestrial Plants and Pest Center, and it's part of a larger proposal that we are all working on, essentially. So there are three objectives from this proposal. The MTPPC wants to know what is the genetic diversity of knotweeds in Minnesota, what species and hybrids are in the population. Are Minnesota knotweeds spreading asexually or sexually? And what markers for herbicide sensitivity and tolerance will a genome-wide study of invasive traits reveal? So what Dallas has been doing covers the first question. However, what I'm concerned with is the second question. I'm focusing basically on the phenotypic traits of knotweeds itself. And a different PI, Roger, is studying the herbicides component on the bottom part here. So I'm looking at basically key invasiveness traits through field and lab studies. Are knotweeds spreading asexually or sexually? And with that, we can go into my experiments. Measuring key invasiveness traits. So this will include all four of these components: adventitious rooting, biomass allocation, pollen viability, and seed germination, both in the field and optimal as well. But before I can get started on my experiments, we kind of need plant material. So we basically got plants from all over the state, as well as a bit from Wisconsin. We collected rhizomes from these areas and got 157 different genotypes from these areas. Populations come all over from St. Paul campus. We have several populations of knotweed right here on campus at the student center, at the gym, at the PGF, at Borlaug. And the Wisconsin population is specifically our source of giant knotweed. We couldn't find giant knotweed in time before all this kind of started rolling. Dallas did find a population of giant knotweeds eventually during her research in Minnesota. And these were all brought here and potted up and added with, so we added substrate and fertilizer, and these were watered daily. And these basically became our source of cuttings and seeds. And these are growing out in the field right now, actually, in the tree nursery. The tree nursery is a nursery located right on St. Paul campus under the Minnesota Agricultural Experiment Station. A very short walk from the plant growth facilities. If you notice like a giant area of trees, that's basically where this is currently happening. So with that, we can kind of start talking about my experiments, which there are four. And as I mentioned before, the 4 points I mentioned is basically those experiments. So we can start with an asexual reproduction experiment, which is adventitious rooting. So here we wanted to know how efficient at rooting are knotweed plants, and do certain species root more efficiently than others? Here is User, one of our undergrads in the Smith lab, with some of our cuttings in the mist house. And on the right here is a picture of a rhizome with roots. They can get pretty big, but so basically all of this is rhizome, and all the fine hairs are roots. All right. And here our hypothesis was knotweeds are very efficient in rooting and creating new knotweed plants. So for the design itself, we basically obtained 10 cuttings from the stock plants I told you about earlier, up to 10 cuttings. Some of them didn't grow well enough to have that many cuttings, unfortunately. We recorded the mass and length of them and standardized each cutting to make sure that there were enough nodes for every single cutting, essentially. So giant knotweeds tend to have bigger internodes compared to Japanese knotweed. So we want to account for the difference in size with using the number of nodes and making that the same for all of them, essentially. These were kept in perlite for 4 weeks and then eventually evaluated for how many roots grew out of them and the longest root length from that. And afterwards, they were potted for biomass, so for the second experiment. And here's just some preliminary data. And here's McKenna holding up a tray of knotweed, if you can see. On the bottom here, that's basically all the roots that kind of came out from that. They were sitting in a tray of water for the entire duration, so they can stay hydrated and not dry out. And on the top right here is a knotweed cutting being evaluated. So you can see grows up pretty substantially within a month. And very, very preliminary data from 2 years ago, where 63 cuttings survived, and the mean number of roots was around 30, but the mean longest root length was about 101 milliliters. And that about wraps up for rooting for now. So after the cuttings were made, we kind of used them for biomass, which is the next experiment, biomass allocation. And for this, we wanted to know how much above and below ground biomass do knotweed produce in 2 seasons, and are certain knotweed species more efficient at producing above/below ground biomass than others. Here's just a picture of the cuttings that kind of grew. This was this year, so they grew pretty big, obviously. And our hypothesis here was knotweeds can produce tremendous amounts of above and below ground biomass in 2 years. For the design of this experiment, we basically used the cuttings from the last experiment and put in pots, about 3-gallon pots, fertilized them, added substrate, and watered them every day, and basically waited for them to grow. So this is for 2 seasons of growth. And during this time period, we would take 2 above ground biomass measurements and 1 below ground after 2 seasons has been done. And we will obtain fresh and dry weight from them. And this is important because we want to know how much plant material they can kind of produce within 2 years. And so the above ground biomass were stored in paper bags after they were harvested and weighed fresh, and then dried in a 95 to 140 degree Fahrenheit oven and weighed again for dry mass. But below ground is a little bit trickier. So it's pretty hard to remove substrate from knotweed roots themselves. They like to be pretty tangled up in there. So instead of just washing off all the substrate and fertilizer and then weighing that and drying and weighing that, we're actually going to be weighing these rhizomes at fuel capacity in the pots themselves. So making sure the entire pot is full of water, and then weighing that, drying the entire pot in the oven, and then weighing that again to get a weight of how much biomass how much rhizome biomass was in the pot itself. And with that, we have covered basically all of the asexual spread traits that I will be measuring for my PhD. So let's move on to the sexual spread portion, which is going to start with pollen viability. So for pollen viability, we want to know how viable are these pollen, and do certain species of knotweed have higher pollen viability than others. So here's just a picture of knotweed with a lot of seed. This is a female plant. And our hypothesis for this one was knotweed plants are highly fertile and can produce high amounts of viable pollen and seeds. So for the design of this study, we basically visually identified male/female flowers. You can pretty much tell from a visual glance, even though they're pretty small, whether the flower is male or female. The female flowers have a much more pronounced pistil and very reduced anthers, and opposite is true for male flowers. So after IDing that, we basically took a step further and looked more closely at the male flowers and assessed the pollen of these. So these were stained with toluidine blue to basically hide the starch in the pollen grains. And we count 100 pollen per genotype, and did this 3 times. So each genotype received 300 counts minimum if they had that many pollen during that season. On the left here is a male knotweed flower, as you can see. The pollen all kind of out here, but the pistil is kind of pretty reduced here. And on the right is a microscope slide of some pollen stained with toluidine blue. The 5 dark blue, pretty well stained, circular circles are viable pollen. The one on with the arrow pointing it, there is an inviable pollen. So we basically did a 0 to 1 yes or no kind of analysis on or count on the pollen itself. Anything that is not a giant circular full blue pollen was considered inviable in our case. And here's just some preliminary data from last year's counts. Yes. So as you can kind of see here, there's pretty wide range of viability in terms of pollen, at least, ranging from less than 25% viability to almost 100%. So knotweed flowers do have can vary a lot in terms of how viable the pollen can be, at least in terms of what we've been looking at. And this is important because we basically the more viable pollen a knotweed plant has, the more possibility it can to basically produce viable seeds, which is important. Plants that have pollen on this end might not make as much viable seed as the one on the left over here. And with seed set, we can basically move on to seed germination as well, which is the last experiment. And this one is a multi-part experiment because there's a lot of components to it and a lot of moving parts. And so we had a lot of questions with this particular experiment. We want to know how viable the knotweed seeds are. Can the seeds survive in Minnesota, especially with the coldness here, the freezing temperatures? Do the seeds require stratification to germinate? And do different knotweed species germinate better than others? And also, do different sizes of knotweed seeds germinate better? And her hypothesis for this was knotweed seeds are highly viable and most can establish into full plants. For the first design, there's 3 designs in this particular case. This is a field germination trial. So we basically planted trays of knotweed seeds, as you can see on the bottom left here, out in the field in Grand Rapids, St. Paul, and Waseca. We took the 5 best germinating in the lab genotypes and basically put them all there, 1,000 seeds in each tray by weight, and did 2 reps each. So 10 trays went into each of the 3 locations. And each of them were counted destructively. So we removed them as soon as we counted them to not confuse with other growth. And you can see Grand Rapids sits a different hardiness zone compared to St. Paul and Waseca. I didn't get into the nitty-gritty 3A, 3B, 4A, 4B splits for Minnesota. But basically, 2 of the cities are in the same zone compared to one another. And here's what they look like in the ground and when they germinate as well. So on the left are my trays up in Grand Rapids. This was taken back when I first planted them in last November. And on the right, here is a sample tray from over here in St. Paul, where a little hard to see, but the circles basically indicate there's one knotweed seedling over there. And preliminary data for this. Basically, Grand Rapids didn't really have very much germination compared to Waseca and St. Paul. St. Paul and Waseca both had some genotypes in the hundreds of which germinate in the hundreds. Grand Rapids only had less than 12. This is average throughout all the different trays. But basically, growth seemed to be stunted. We're going to repeat this experiment this coming year as well to see if this is a trend that continues. But that's kind of the takeaway. It seems to germinate a little better in hardiness zone 4 compared to hardiness zone 3. And with that field experiment out of the way, we can talk about the optimal experiment, which is design part B. So we basically took 7 to 8 genotypes, some of the best germinating ones in our collection of seeds, and treated them differently based on different treatments that we thought of. So one control, just full light, nothing to do with them. One was covered in darkness for 4 weeks. One was put in a cold room for 4 weeks. One was frozen for 24 hours and then taken out. And 3 treatments of water and 2 gibberellic acid concentrations as well. So gibberellic acid is known to help stimulate germination in some seeds. So we want to see if that helps basically stratify and help them germinate, essentially. So for these, we did 200 seeds per rep and did 2 reps per genotype. And these were also distributive counts as well. And they were observed over 4 weeks too. Here's what they kind of look like in a very long tray. And a lot of counts as well from all different trays and different treatments. And the last study basically is the same as the previous, except this is comparing seed size now instead of the different treatments. So no more light and darkness and GA and water, what have you. So the exact same as before, but basically just split up 7 genotypes based on small, medium, large seeds and observe them as they germinate in full light. And here's just some preliminary data from that experiment. So you can kind of see small seeds might not be the best option for some of these genotypes, but this is very preliminary data. I wouldn't take this to heart. This is just one observation in one season. But medium and large seeds seem to be doing okay for the most part across all species, or yeah, across all genotypes. And that's basically it for the experiment. So any directions we can move forward in the future. It's probably wise to combine my data with what Dallas has done as well in a genome-wide association study to kind of maybe link phenotypic traits with genotypes. We can also consider doing a seed to plant study. So observing plants germinating from seed all the way to an entire plant, see how invasive they can be, if they can reproduce asexually or sexually, and maybe also a seed burial study. So how long can knotweed seeds last in soil or substrate? It'll be interesting to see if they can last 5 years, 10 years, 20 years, etc. But there's obviously a lot of different directions this can kind of take. So with that, I would like to thank my committee, Alan and Neil, my advisors, as well as Roger, Matt, and Sue in my committee. The PGF staff, Pam, Todd, Dean, and Doug. I couldn't have done a lot of this work without all their help. And Joshua from Crop Services as well. In the Smith Lab and Anderson Lab, Marie, Dallas, User, McKenna, Grace, Sarah, and Cora. And from both research centers that we collaborated with, Dan, Keith, Rosmanita, and Crystal from the North Central Research and Outreach Center. And from the Southern Research and Outreach Center, Jason, Forrest, and Charlie. And especially a big thanks to the Minnesota Invasive Terrestrial Plants and Pest Center for funding this research. And with that, I will now take any questions.
Thank you, Kevin. If you would like to ask a question, please put it in the chat or in the Q&A. And if you're in the room, please feel free to go ahead.
Go ahead, Emily.
Can you tell me, I was interested in Bohemian knotweed, and it has so many different voiding levels. Does
Yes.
that impact the seed set, pollen viability?
We don't know quite yet. Actually, so Emily asks if quality level of Bohemian knotweed affects seed sets, and what was the report? Yeah, seed set. We don't quite know that yet, but we are looking into the poly level in a side project, and one of our undergrads that's actually doing a Europe on poly level in knotweed. So we will know more once she's done her analysis and can compare that with our seed set specifically. We aren't actually looking at seed set per se, actually. So that would be something interesting to kind of consider after was work has been done for Bohemian knotweed, and someone can compare that data with seed set, I guess, in those plants. But we don't know at this moment.
I was curious, what were the environmental conditions in Grand Rapids, and how were they different from the other two sites?
Well, Grand Rapids is a lot colder than both St. Paul and Waseca. It sits at a higher zone, and conditions otherwise weren't too different besides climate, I believe. See, Grand Rapids. So both Grand Rapids and Waseca are research stations. So the soil is kind of, tended and planted with stuff regularly. So they're similar in that sense, but beyond weather and latitude, there's not too many differences, I guess. Yeah.
Can you tell us a little bit more about knotweed? Like, so for instance, like what kind of fruit does it produce? And like from one of those flowers, are we talking about thousands of seeds per plant? Because you're dealing with a lot of seeds in your experiments.
Yes. So one of my slides actually mentioned it. One knotweed plant can produce thousands upon thousands of seeds, and they're very, very small. They look like sesame seeds, kind of, but they're wrapped in basically a papery kind of winged triangular kind of shape. I probably have one on here. Let me see. But they're very tiny. They can basically be transported through air or through water. Let's see. I had one of my slides, but yeah, here we go. So that's my hand. So they're very small. But basically they're achenes, so they are individual seeds. And the white part you can kind of see, that's the wing. The entire thing is called samira, and the wing papery part basically helps them transport.
We have one question in the chat from Matthew Clark. What should people do if they find knotweed around the state?
Around the state, depending on. So the question was, what should people do if they find knotweed around the state? It really depends on like what property it's kind of on, I guess. If it's government property. If it's government, then the government, they kind of have to deal with them themselves. If it's on someone's property, then they might be interested in kind of removing that from the property. So it depends on location, first of all. If it's just out in the wild, that might be more of a government thing, depending on zones and stuff. But if it's their property and they want to remove it, then they can, like I said in the previous slide, try to remove it mechanically and also use chemical controls to remove it. So you basically want to cut off all the stems first to expose the rhizomes. And then glyphosate works pretty well in destroying the plant completely by applying it to the exposed stem and having it travel down all the way the rhizomes throughout the entire plant to kill it. Otherwise, you can try digging up the entire thing itself, but it depending on how big it is, that might not be feasible. But that will be the best option to remove the entire plant from whatever area it's currently in.
Okay, along somewhat the same lines, are knotweeds still sold as ornamentals? If not, are there any species that are lookalikes or not a concern? Not a concern?
So knotweed is. The question was, are knotweeds still sold as ornamentals, or are there similar species that are lookalikes? Yes, there are some species that kind of look like it. So another name for knotweed is fleece flower, and there are fleece flowers being sold, but they are not as invasive as knotweed, of course. But to answer your question about whether they're being sold or not, so they are on the list of species that people can't sell, basically. But we were able to obtain a variety species, Philopia japonica or compacta, which is a smaller species of knotweed you saw in the very first picture with pink flowers. That is not easy to find, but it's sold somewhere. I'm not sure exactly where it's sold, but that's still being sold somewhere. But in the States, at least in certain areas, it's prohibited from being sold, essentially.
And we have a comment from Roger Becker.
Mhm.
Nice presentation, Kevin. Could you elaborate more on the pros and cons of needing to measure root biomass in pot?
In pot. So Roger asks, can you elaborate on the pros and cons of measuring root biomass in pot? So I'm not entirely sure what he meant by that, but there's limitations to measuring it in the pot itself versus out of the pot. It's an approximation weight compared to, removing all the substrate from the rhizome itself and measuring it that way. But yeah, so it's basically very time consuming to remove every single little particulate from every single substrate particulate from the rhizome itself, compared to an approximation weight, which might not be as accurate, but will give us a pretty clear and faster picture compared to washing the entire thing by hand slowly, because we have a lot of reps to kind of go through as well.
So I have a question. So this is the diversity plot you showed.
Mhm.
PCOA plot.
PCOA, yeah.
What does that actually mean?
So
You described what the plot's so totally, but what does it actually mean as to your own work?
So the question was, what does the PCOA plot, what Dallas has done, relates to what I'm doing? So basically, Dallas's work helps with answering one of the objectives in the proposal for this entire research. What I'm doing is a different objective from that research, but still on a similar line. Because I'm looking more at the phenotypic traits of knotweed, so biomass, seed set, that sort of stuff. Dallas is more concerned with the genetic diversity of knotweed and what kind of knotweed is found where in Minnesota specifically. So similar lines, but if we combine both what Dallas and I did together, we can maybe identify regions where it might explain why certain species might be demonstrating certain traits. Well, more questions? If not, then
I'm not got a feeling that, I'm really curious about this one. So you're doing a seed size experiment.
yes.
Do you know that those small seeds are viable? Because if you look at your germination percentages, it may not have anything to do with seed size, it might have everything to do with non-viable seeds.
Well, let's see. So we did test the seeds beforehand to make sure they actually germinated first before we did all the studies. So we do know that they do germinate, but how much of them are viable or not, we don't know that, of course. But yeah, so we basically tested the seed before, so we know that at least some of them do germinate. We just don't know like if seed size is a factor in that or not.
So really comparing small, medium, and large within a genotype is probably not as important as looking at seed size across.
Yeah, we did that. We are doing that.
The way you're presenting the data is you have these stats.
Yeah, it wasn't the best graph, I think, for it, but
Definitely.
well, if there are no any questions, I will end the presentation here. Thank you all for your time. And oops, stop sharing.